The components of nucleotides include nitrogenous bases such as adenine, thymine, guanine, and cytosine, a deoxyribose, a phosphate group, and make adenine, thymine, guanine, and cytosine. The DNA double helix was discovered in 1953 by James Watson and Francis Crick. It was discovered by beaming X-rays through DNA crystals and then recording the scattered X-rays with photographic film. It created a cross shape, which is common for a helix-shaped molecule. A hydrogen bond is a dipole-dipole attraction that forms between two hydrogen bonds, which allows the nitrogenous bases to bond, forming the rungs of the twisted ladder known as the DNA double helix. The strands on a DNA double helix run anti-parallel, which means they run in opposite directions. Step 1. DNA double helix is unzipped by helicase, an enzyme that breaks the hydrogen bonds holding complementary bases of DNA together. Separation of DNA forms replication fork. Step 2. An RNA primer is attached to the end of the leading strand. Several are attached at random points on the lagging strand. Step 3. A small piece of RNA called a primer binds itself to the end of the leading strand. It acts as the starting point for DNA synthesis. DNA polymerase binds to the leading strand and moves across it, adding complementary bases. RNA primers are made and bind to random points on the lagging strand. Chunks of DNA known as Okazaki fragments are added to the lagging strand. The Okazaki fragments will join up later. Step 4. After all of the bases are matched up, an enzyme called exonuclease strips primers off of strands. The gaps where the primers were are filled with more complementary nucleotides. Step 5. DNA is proofread to avoid mistakes in the sequence. Step 6. An enzyme called DNA ligase seals up the strands into two continuous double strands. The result is two new DNA molecules with one new and one old strand of DNA.
Transcription of DNA to RNA. Differences between the two. Here's a model of DNA and RNA. So one of the differences between RNA and DNA is that DNA is two strands while RNA only has one. DNA is much longer than RNA. RNA is actually pretty short. Another difference is that DNA has deoxyribose sugar while RNA has ribose sugar. Another difference is in the nucleobases. In RNA, there is uracil. Meanwhile, in DNA, there is thymine. Yet another difference is that DNA makes RNA, makes proteins. Different types of RNA. There is tRNA, which is transfer RNA and carries amino acids to ribosomes. There's mRNA, which is messenger RNA and tells the ribosomes what to make. There's rRNA, which is ribosomal and helps the mRNA bind and can also act as an enzyme to speed up reactions. So basically all of this happens in the nucleus. Introns and exons, yay! Basically here in the RNA, the introns get cut out because they don't work with this, and then the exons, which are the remaining parts, get glued together. Making of mRNA message and the leaving of the nucleus. For replication of DNA, it must first get unwound, which happens by an enzyme coming along and straightening it out. After that, we unzip it with the helicase, which is that enzyme. The promoter tells this where to happen, and the RNA polymerase is the thing that makes the bases match up. At the end of this, there's a terminator, which tells the enzymes to stop working. Then to finish this off, you just add a poly-A tail and a 5 cap.
Genetic code and how to decipher it. Genetic code allows DNA and RNA to be decoded into proteins. It's decoded in groups of 3 called codons and specify an amino acid. Protein creation starts with a start codon and ends with a stop codon. After the third stop codon, the protein is made. The reading direction depends on the ends of the DNA or RNA, which are the 5' end and the 3' end. Protein creation starts with initiation, which start with the mRNA ribosome and an initiator tRNA gathered by the initiation factors. Small and large ribosome subunit envelop the mRNA to start translation, which matches corresponding amino acids to create a polypeptide. The tRNA carrying methyl 9 attaches to the small subunit binding to the 5' end of the mRNA. The will move towards that 3' end until it reaches that start codon. We are now onto elongation. Inside the ribosome are 3 areas, site A, P, and E. So ape, very much ape. The first methylline carrying tRNA starts in site P. Site A will have an anticodon that's complementary to the first. rRNAs inside the ribosome creates a reaction and the 2 amino acids connect, turning into a peptide. It also transfers the methylline from the first tRNA to the amino acid of the second. The mRNA is pulled forward by 1 codon, allowing the now empty first tRNA to exit to site E. A new codon is exposed in site A and the process repeats. That is, until termination, where the finished polypeptide is released. That happens when a stop codon enters site A. It is recognized by proteins called release factors that adds a water molecule to the last amino acid of the chain, thus separating. Afterwards, the small and large ribosomal subunit also separates from the mRNA from each other to take part in another round of translation. Meanwhile, the newly released polypeptide pulls together and creates a protein with a mustache.
3 types of mutations are frame shifts, substitutions, and inversions. A frame shift mutation is a mutation such as an addition or deletion that changes how the codon sequence is read. A substitution is when one nitrogenous space is replaced with another nitrogenous space. An inversion is when an order of nitrogen spaces are inverted to create a different order. Some mutations don't affect the structure and function of a polypeptide, which are called silent mutations, but others cause the non-functioning polypeptide with a different structure. Silent mutations are mutations that have no effect on the structure and function of a polypeptide.
Mutations in albinism. Albinism is a frame shift mutation. This is the normal process. The polypeptide created is pretty long. This is the mutated process. The polypeptide created by the mutation is much shorter. When made into a protein, the shape is weird and it can't function properly, thus resulting in pale skin, eyes, hair, and susceptibility to sun. Yeah, you learned a thing.